Hi, everyone. It's my pleasure today to introduce Dr. Kathleen Morrison, who is visiting us from the University of Maryland School of Medicine. And she received a dual bachelor's of science in both biology and psychology from West Virginia University in 2006, so right around here. She earned her PhD in biological psychology from the University of Tennessee in 2012, and she started a postdoctoral fellowship at the University of Pennsylvania with Tracy Bale in 2012. Oh, sorry. She started a postdoctoral fellowship at the University of Pennsylvania with Dr. Tracy Bale in 2012, and then recently moved to Baltimore when Dr. Bale transitioned there. She's published 15 papers, more than half of them as first author, and in such journals as Biological Psychiatry and Nature Neuroscience. In her research career, going all the way back to her undergraduate days, she's been studying the neural mechanisms of stress-related behavior, and her most recent work is highly translational and involves studies in both humans and rodents. In 2017, she was named Associate Director of Translational Research at the Center for Epigenetic Research in Child Health and Brain Development at the University of Maryland School of Medicine. In addition, she has recently been awarded a prestigious K99 R00 on the role of hormonal state and programming effects of peripubertal stress in females. And we're very excited to have her join us today and tell us about her work. Please give her a warm welcome.
Great. Thank you, Georgia. That's a lovely introduction. And thank you all for being here and for inviting me to come talk to you today. I'm really excited to talk a little bit about what I've been working on in my postdoc. So it's probably not news to this audience, but I always like to start out with why do we care about neuropsychiatric disorders? And this is just one example of the burden of neuropsychiatric disorders. And this is from the United States. And so this is daily, so disability adjusted life years. So this is the total number of illness years. Yes. Total number of years lost to illness, death, or disability. And you can see that neuropsychiatric disorders are #1 on this list. So they're a huge burden in our society. So of course we want to understand how to treat them and what's their etiology. So I've really been interested in what puts individuals at risk for neuropsychiatric disorders. And in my career, I've studied stress as a key risk factor. So exposure to chronic, extreme, or unpredictable stress throughout a lifespan puts an individual at risk. Now, I always do like to point out that if you take a group of individuals, so when I talk about these data with non-scientists, they're like, oh, I'm so stressed, like you should study me, right? And like we all do experience a lot of stress, but we know that about 20%, depending on the neuropsychiatric disorder, actually precipitate into disorder, whereas other individuals remain in a non-disordered state. And so I've really been interested in if we all experience stress, and some people will experience the exact same stressful experience, what pushes an individual one way or another? And when we think about what those factors are, it's helpful to conceptualize them in the G by E by D. So your genes, by your environment, by the developmental stage in which you experience this exposure, can really push you in one direction or another. And so that's kind of an approach I've taken in my research. And another big factor that doesn't kind of, in the G by E by D that influences all of them is your sex. So in each of these instances, the sex of the individual will influence all of them. And so that's one of the reasons I actually joined Tracy's lab, to gain expertise in sex differences and how that influences neuropsychiatric disorder risk. Another reason I joined was because in my grad school, what I was doing, and what largely I think the stress research as a whole, aside from some notable exceptions, are studying males, right? So animal models focus on males, but that doesn't capture female risk. And so why do we focus on female risk instead of just adding females to male-focused studies? And one of the reasons to focus on women is that we have this disparity in what types of disorders women are likely to get compared to men. So you see that things like affective disorders, anxiety, depressive disorders are greater in women in prevalence, whereas neurodevelopmental disorders like ADHD, schizophrenia are more likely to be found in men. However, we're not necessarily studying the risk factors that are relevant to women. All right. One of those big risk factors comes in the developmental stage. So a lot of work on stress and the lifespan is focused on really early life, where males tend to be at higher risk. What we see from the epidemiological literature and from some animal studies is that puberty is a really important vulnerable time for females. And so early life adversity in the years kind of surrounding puberty onset is one of the strongest predictors of affective dysfunction in adult women. And periods of hormonal flux, like pregnancy, postpartum, or menopause, exacerbate the risk for affective disturbance. Okay? So when I joined the lab, the goal was actually to build a mouse model relevant to female life experience. So not to just take adult females and study them beside our males, which is a perfectly good approach, but to actually ask, can we understand the lifespan of a female and those factors that put her at risk? And so when I joined, we were funded by a center grant. So this is on the left, Neil Epperson, who's the clinician that we worked with. And then on the right is my mentor, Tracy Bale. And so with the center grant, we started this Penn Promotes Center and to set out to build a translational study side by side from the beginning. So to recruit the human subjects and to design the mouse model together to make it as translationally relevant as possible. Now, Neil has also left Penn. She's at Colorado now. At the medical school there. And again, Tracy's founded the Search, the center at Maryland. But we continue to, kind of forge these collaborations. So in designing this, we focused on the three main periods of hormonal change, right? And puberty as the period where the programming, the biggest risk for exposure to stress. And then we're looking at outcomes in pregnancy and postpartum, or there's a whole other arm of these studies that where we're looking at aging and menopause. And so in the humans, we use the Adverse Childhood Experiences Scale, or ACEs. So you hear me say ACEs a couple times during the talk. And this is a widely used, well-validated scale that's been shown to, you can have up to 10 ACEs, so there are different categories of abuse, neglect, or household dysfunction. And you can endorse whether you've experienced these and when in your lifespan you've experienced these, importantly. And then we can add up. And through a lot of other studies, there's a Kaiser Permanente study that looked at a wide range of individuals. The more ACEs you have, the more you are at risk for a variety of early death, of cardiac disorders, all kinds of things, including neuropsychiatric disorders. And so this is the framework that we're using for the human studies. As an aside, so we have this whole project on menopause and on aging. And so that was a study I published with so Sneha was a rotation student in the lab, and Ethan was actually a very precocious high school student who contacted us and came in, and he was able to be on that paper, so that was really great. And now he's now graduated from Penn, so, okay. So one of the important things when you're designing a study to be translationally relevant is what is your outcome gonna be that can actually bridge mouse to human? And so we focused on the axis. Pituitary adrenal. So for those of you that are familiar with this, just a quick primer. So the PN, the paraventricular nucleus hypothalamus, is the H, releasing pituitary, which is ACTH, adrenocorticotropin hormone, to the adrenal cortex, where among other things, it stimulates the release of glucocorticoids. That's cortisol in humans, corticosterone in mice. Importantly, as we're talking about see in some of these outcomes, the placenta also contributes to circulation. So think about mouse, a placenta contributing to something we had to consider when we look at our outcomes. On the HPA axis, regulation of this axis is a central endophenotype of affective disorders, so we see it across many disorders. The circuitry is evolutionarily conserved, so we can make assumptions about what's going on in a human by looking at a mouse relatively easily. We don't have to extrapolate too much. It importantly undergoes maturation during the time window we're interested in. So during puberty, the HPA axis is maturing, and so it might be vulnerable to our stress exposure. And we already know that changes in HPA axis responsiveness are associated with, in pregnancy, are associated with affective dysfunction. So there's been a link there. All right, so I'm gonna the way we typically do this in mice, I'm gonna show a lot of graphs like this today, is we restrain them in a conical tube. It's very simple. For 15 minutes, it's a very brief stressor. And what you see in any individual, human or mouse, when you stressful exposure, is you'll see glucocorticoids rise and fall over time. And this is, I always like to point out, a completely appropriate normal response. This is what you're supposed to do when you experience a stressor. So we're not saying the HPA axis is actually important. It's when it or too low, then we see this. So this would be, we do 30 minutes at our peak. So we'll do 15 minutes on the rise, 30 minutes on the peak, and then we do 120 minutes out for recovery. Mm-hmm. Okay. So we first wanted to know what we're trying to model in humans, right? And one of the early outcomes that we were seeing was on a maternal infant separation stress. So our clinical collaborators recruited women during pregnancy and asked them about their adversity that they had during puberty. And then one of the tests they were doing was this maternal infant separation test. And they were actually interested in the babies, to be honest. So they wondered, what mom has, how does that affect the babies? And so they were taking the babies in the other room and do this brief restraint stress on the babies, and they're getting saliva from them. But in one of our meetings, they happened to say, you know, we can tell who the high ACE moms and the low ACE moms are from how they respond to this test, just by watching them. And the high ACE moms don't seem to respond. They don't seem as upset that their baby's upset. And so we asked them to take saliva from the mothers so we can look at that as well. So this is what they did. It's a very brief separation. So we have baseline saliva collection and then 4 time points after to look at their cortisol. All right. What we see is that high ACE moms have a reduced cortisol response to this infant separation test, okay, like we actually had predicted, which is kind of amazing. So another question I always get about a low response, like, isn't low better? Or does that mean you're not stressed? Like, isn't that great? That's not the case. It's important to have an appropriate response, but we also contextualize these in other tests. So they assessed Edinburgh postnatal depression scale in these women to try to give this some context. Are they, you know, how are they reacting? And they score significantly higher, the high ACE moms, in the Edinburgh postnatal depression scale, suggesting that in context, this is not a positive outcome to have a lower cortisol response. And they're just not responding to their babies. I mean, they saw that visually. They just weren't responding to their babies being stressed. Okay. So then when we're designing our model, the hypothesis was that puberty stress, so stressing these animals when they're young, when we grow them up and we have that experience interact with the dynamic hormonal changes of pregnancy and postpartum. So we're using this as kind of a natural way to manipulate the hormonal state in a really naturalistic way. That when those two things combined, we'll see a disruption. And we predict it will be lower based on what we see in the humans. Right? Right. So the experimental design, puberty in our mice, we use a mixed strain, C57-129 strain. Do 2 weeks of stress from postnatal day 21 to 34. On each day, they experience 1 of 7 stressors, so this is a chronic variable stress, so they don't habituate to the stressors. None of them are extreme. The most extreme might be exposure to fox odor, a predator odor. That's just pretty extreme. But otherwise, we're disrupting their circadian rhythms, we're restraining them, and it's all unpredictable. And then we let them grow up, and so importantly, they are singly housed for the stress exposure, so they don't have any way to any kind of potential coping that might happen, or they are singly housed at postnatal day 21 through 34, but then we pair house them following that because we don't want to study the long-term effects of social isolation. We know there are long-term effects of chronic social isolation. All right, so then in pregnancy and times around pregnancy, we assess separate cohorts of animals for their stress response. And so again, this is a time where we're using this naturalistic change in hormones. So even early pregnancy to late pregnancy, we get a different hormone profile, and same postpartum. So we're able to look at all these different time periods, including estradiol, progesterone, the big ones, but also things like allopregnanolone, which is a potent neurosteroid and is changing throughout pregnancy. And again, we give them this 15-minute restraint stress and assess their response. Right. So both nulliparous, so these are adult, non-pregnant, they've never been pregnant females, and actually postpartum, we didn't see a significant effect, right? So here we have corticosterone on the y-axis and time on the x-axis, and the gray bar just indicates when they're in the restraint tube. And after they come out of the restraint tube, they're just freely moving around their cage. And kind of amazingly, this was amazing to me, it's still amazing to me, that either early pregnancy or late pregnancy, we see, as in the humans, a significant reduction in their corticosterone response to this restraint stress. So this was very exciting as a new postdoc to kind of find something that we felt we had some traction and a mouse model we could work with. Now, one of the things that bothered me was this postpartum finding, right? So in the humans, the test was postpartum. And one of the things we know about postpartum, for one, glucocorticoids are very low, so you're kind of dealing with a floor effect. But also the brain's really changed to focus on maternal relevant things. And so we designed a maternally relevant stressor, just like they did in the humans. And so I took a mouse cage and I bisected it. So it has a mesh barrier so they can see them, smell them, hear them, but they can't retrieve them. And if you don't know anything about mice, they like to bring their pups back into the nest, right? They'll do that. You spread them out, they'll bring them right back. And so instead of the restraint stress, we used this 15-minute separation where they couldn't get to the pups to see if that would elicit any differences. And it did. So we need this maternally relevant stressor to see a reduction in the corticosteroid response to this stressor. We also looked at their behavior during the task. Unfortunately, the cage wasn't quite big enough. This needs to be kind of modified and expanded upon. But we saw a slight reduction in the distance traveled and some kind of inklings of that the CVS females didn't spend as much time here at the barrier, although nothing significant. But when we correlate these two measures, so with the idea that increased glucocorticoids might be increased arousal, would be increased distance traveled, so we might expect a positive correlation, which we see in the control animals that's disrupted in the CVS animals. So there's some disruption of their stress responding. Okay. And so now we have this working model where stress during puberty, when interacted with the unique hormonal state of pregnancy or postpartum, leads to an altered HPA response. So then the question is, what tissue is being reprogrammed by pubertal stress? And I say what tissue because as a neuroscientist, I was like, of course it's the brain. But if we go back to the HPA axis, there are lots of parts of that aren't the brain and could easily be changed by our stress. So this is definitely one of the things I've learned in Tracy's lab is it's not all, it all interacts, right? So we looked, long story short, we looked in the pituitary, the adrenal, and the placenta on gene expression, on functional measures. We also looked at circulating hormones, and we didn't see a difference. All right, so we pretty much ruled out any major effects in the peripheral part of the HPA axis. So the neuroscientist in me said, all right, great, now we can start looking in the brain. And when we're looking in the brain, we pick the most proximal part, right, the PVN, and is the programming. Is there anything changing there by pubertal stress? And so what I did was took the PVN from late pregnant females and performed RNA sequencing, right? So now we're looking at the transcriptome. This is all the mRNA that's available in the PVN at the time of sacrifice. And based on my lab, we have other people in the lab who do this, and they get it's like amazing, beautiful heat maps and 1,000 differences, and I had 22. So for those of you doing RNA-seq, if you're thinking about that in the brain is a very heterogeneous tissue, and you can wash out a lot of effects because you have, especially places like the PVN, many different cell types, and a gene can go in opposite directions, so there's a lot of noise. And there are now things like single cell, you can do things like ribotag, where you can pull down just transcribed mRNA. There are ways to get around that in the brain, but I was in a seminar once that a bioinformatics person, and they were like, oh, don't do the brain. I was like, that's all I do, so you gotta help me. Anyways, so we, working with what we have here. Among those genes, there were 6 that were of interest to me, and that's because they're all immediate early genes. So these animals were taken at baseline. There was no stimulation. So I thought this was really strange that in the CVS females, we have increase in all of these immediate early genes, which canonically shouldn't be on at baseline. So it's kind of hard to formulate a hypothesis around immediate early genes. They target a lot of things turn them on. But what it suggested to me was that the chromatin state of these animals was inappropriate. Why should these genes be available for transcription, right? Perhaps this signals that the chromatin's more relaxed, so the DNA is more unwound, and the genes are available to be transcribed. So that was the hypothesis I generated out of these data. And in between doing this and kind of starting the project, a new technology, kind of sequencing technology, came along that was actually really useful for my question, and that's called ATAC sequencing. So ATAC stands for Assay for Transposase Accessible Chromatin. And so the chromatin, so the DNA is either wound up in chromatin and it's closed right here, and so you're not going to get genes are not going to be transcribed, or it can be open. And what happens is you add these transposases that go in and they'll cut the DNA where they can find it. And so they'll cut the DNA, and you can get transposases that have, they're ready for sequencing. They have libraries on the end, so you can throw them in the sequencer. And then we can ask is the chromatin more open? So this is a great way. I was really excited when this came out. This directly addresses my hypothesis about, is the chromatin more open? And if it is, what's it telling us? So the first thing, I took females that had been stressed during puberty, and again in adulthood, so either pregnant or we also had non-pregnant females for this assay. We take the PVN, we sequence it, so we're very lucky to have a sequencer in our lab, so we can do all of this from animal to bioinformatics. We do this in our own lab. We can do the sequencing and then the analysis to answer our question. Okay, so if we just look at non-pregnant females, right? So the idea is that puberty changes something that's lasting into adulthood. We should be able to pick that up somewhere. We don't see it phenotypically. It seems to be sub-threshold, but are there any differences before they even get pregnant? And there are. So this is a heat map. So each column is a different peak. So these are not necessarily all intergenic. So if you do something like a TAC-seq, you're getting a lot of outside of gene coding regions. But each peak, each of these is a differentially expressed peak, and the rows are different animals. So these are individual biological replicates in control and CVS females. And what we see is red indicates more expression. And so we have increased expression, so more openness, more reads on these peaks in our CVS females, which is what we predicted based on our RNA sequencing. So this was exciting. Now you can take all these peaks. So this is like really pretty, but it doesn't tell you much, right? So you can do things, functional annotation analysis to say, okay, do these match up anywhere? Do they make sense? And when we do that, we see that we get, so these are functional categories that these genes have been annotated to. And we see things like synapse, sodium transport, calcium ion binding, among other things that might suggest that the PVN's in a different state to respond to input now, right? So we're slightly changing something, this latent kind of reprogramming that's gonna poise the PVN to respond differently to the effects of pregnancy and then when we stress them during pregnancy. Any questions about sequencing or texting? Okay. All right, so then what happens when we look at them? What does pregnancy do? And this was an important consideration because in some of our assays, we do a lot of things surrounding pregnancy. Pregnancy just wipes out the effect of anything else. It is such a major wholesale change on the body and the brain that you can just, stress during puberty, like no big deal relative to what pregnancy is going to do. So I was concerned that we wouldn't see anything at all. Now, we did this analysis. You can do these kinds of analyses two ways. You can set a p-value cutoff and look for differentially expressed, but it also can be helpful to look at things in an unbiased way. So one of the ways to do that is called rank-rank hypergeometric overlap. And again, with a tissue like the brain where it's very heterogeneous, this can be helpful. So what you do is you can say, what's the effect of, so you compare virgin, in this example, virgin versus pregnant controls and virgin versus pregnant CVS females, and you include all of your peaks. So you're not cutting anything out based on p-value. And then they rank them from most highly expressed, upregulated to downregulated by your treatment, in this case pregnancy. And then you can match them up next to each other. And if you have a perfect match, so pregnancy does the exact same thing to controls and CVS females, you get a bright red line down the middle. And you know that if you put the same data in twice and you make a mistake and you go like, oh, that's how it works. Okay, same bright red right across the middle. It's a good confirmation. But what we see here is not a lot of red. There's not a lot of overlap in what pregnancy is doing in the PVN of control and CVS females. So like we've seen with the HPA axis, what pregnancy does is actually pushes these two groups further apart. So it enhances all the phenotypes and drives them away from each other, which we're seeing here also in the chromatin state as well. So when we do apply a p-value cutoff, you can do that. And so this is the effect of pregnancy either on control or CVS. So these are differentially expressed peaks by pregnancy. And then we can ask, are they the same, right? So it's another way to kind of answer the question that the RRHO was answering. And you can, again, do functional annotation clustering. And like maybe the peaks aren't the same, but the pathways are. That's always a possibility, right? Like the pathways are basically the same. It's kind of slightly different genes that are doing it, but in the end result, it's the same. And we don't see that here. So 3 of the top, see these are all significantly enriched in our subset, and 3 of them are the same. So we get synapse, actin binding, and transmembrane are all the same, which makes sense there will be some overlap, right? Pregnancy, there's something it's doing to everybody. But otherwise, we see big differences in the kind of functional pathways that we see, including things like, I didn't know things like the death domain existed. It sounded bad, it's bad. It's not good. So in our stressed animals, there are different processes being engaged on the chromatin level here. And so this is exciting. This is fairly new data, so I'm still digging in to see kind of what this means and what kind of predictions we can make out of it. Okay. So now we can add to our working model, right? So we've focused in on the PVN. And importantly, if you've studied the PVN, there's also the peri-PVN, which is important for the function of the PVN. So the peri-PVN is this kind of halo of GABAergic and glutamatergic cells surrounding the PVN that is thought to actually integrate all the inputs. So the PVN is receiving input from a lot of brain regions, and it has to decide, right, go, no go, CRF, no CRF. And it's thought that the peri-PVN acts as a nice aggregator, right, to basically send GABA, glutamate, and tell the PVN what to do. And so that we consider that here in our working model. But we've been able to add that one of the things that puberty stress does is that it changes the chromatin state. It is in a more open state before they're pregnant, and it becomes more different during pregnancy. Now, one of the downsides of the bulk, right, sequencing is we don't know what cell type this is in. And so future analyses would be able to utilize single cell, things like ATAC-seq or RNA-seq, to get at what's the cell type here. Okay. All right. And another really interesting question as part of my grant proposal was, what is it about pregnancy that uncovers this phenotype? So we don't see it before they're pregnant. We haven't seen it when they're aging. It's very specific to pregnancy and postpartum. And what is it here that we're seeing? Now, another clue that came from the RNA sequencing, this is taking you back to the RNA sequencing of the PVN. Another gene of interest that came up, increase in the CVS females, is VGAT. So VGAT is a vesicular GABA transporter. So it brings GABA to the synapse so that it can be released. And like I just said, GABA is really important here in differentiating whether the PVN is going to send a signal in terms of CRF. And so this was really interesting. This was kind of a hint at what might be going on, because there are a lot of accommodations that are made during pregnancy, right? A lot of change to hormone receptors and things like that. So what if it's just GABA? This was a really interesting idea. And one of the reasons it was is because of allopregnanolone. So allopregnanolone is a key neurosteroid in controlling the PVN. It's a positive allosteric modulator of the GABA-A receptor. So now we have GABA is really important here in the PVN. Now we have this neurosteroid that increases in the brain during pregnancy. And so a positive allosteric modulator is that it doesn't do anything to the GABA-A receptor on its own without the presence of GABA. But when GABA is there and allopregnanolone is there, it potentiates the effect. So it heightens the effect of GABA, increases inhibition, which would fit with our reduced glucocorticoid response, right? So this is our hypothesis. And we've already seen that allopregnanolone has been implicated in pregnancy-associated just normal changes in the HPA axis. So it seems to be an important player here that kind of fits with our findings. All right, so for this I took a variety of pharmacological approaches to see if we can figure out if allopregnanolone is key. And so one of the first things I did was just to broadly block the synthesis of allopregnanolone by blocking the enzyme that converts it. And so during late pregnancy, I gave peripheral injections of indomethacin, which does block this enzyme here, and expose them to restraint stress. And what do we see? So in our control females, indomethacin doesn't change their glucocorticoid response. You can see there's no significant effect here. But in our CVS females, so if allopregnanolone is what's driving it lower, if we release allopregnanolone, we should see it going higher, and that's actually what we see here. So allowing there to be increased blocking allopregnanolone allows the HPA axis to respond like we see in controls. So one of the challenges of pharmacological approaches, right, is that no pharmacological approach is perfect. So we have to use a variety to kind of hone in on them. And I actually thought one of the good uses, one of the interesting things to do here would be to use our males, right? So if it's really the puberty stress changes GABA tone, and that it's about seeing allopregnanolone to kind of push that further, are males just never seeing allopregnanolone? So previously we'd never seen an effect in males. When we started this, of course, we were looking at the males as well to see what kind of effects there would be there. And as we predicted, we didn't see anything. So that makes them a nice group to use to kind of do these pharmacological manipulations. And so what we did was actually give them allopregnanolone, so increase their levels of allopregnanolone and then assay how males respond to a restraint stress. And so we gave doses of allopregnanolone that are shown to increase allo in the brain of males prior to their receiving a restraint stress. And another finding that I just couldn't believe, it's great. So in the control males, we don't see an effect of allopregnanolone. They're not sensitive to it. In the CVS males, so again, these are males that had stress during puberty and now they're adults, and we've just given them this increase in allopregnanolone. We recapitulate the blunted response that we see typically only seen in pregnant and postpartum females, suggesting that allopregnanolone is sufficient to do this even in males, right? So this was another kind of helped point the way away from things like all the hormone receptor changes in pregnancy towards its GABA, perhaps, and allopregnanolone binding here. And so then the last study that I did pharmacologically was to do more specifically, right? So the problems with the two that I showed you just now is that they're peripheral, right? That's going to block, indomethacin is going to block allopregnanolone everywhere, and as well as a lot of other hormones. So it synthesizes a lot of other hormones, and the allopregnanolone in the males is also going everywhere because it was peripheral. So here I did a site-specific. So targeting the PVN with bilateral cannula, and then giving a drug that's actually more specific as well. So 17-PA is a drug that blocks the neurosteroid binding site on the GABA-A receptor. And so it's going to more directly address that question of is it allopregnanolone on the GABA-A receptor? And these popped up. But so what we see in vehicles is we see our typical reduction in glucocorticoid response. Now the curve is slightly shifted here because these animals had to get their injection. So anytime you pick up a mouse, their HPA axis starts. So just like FYI. And I learned this the really hard way of doing this on all these animals and saying like, oh, they're already at 15 minutes here. But that's okay, we can predict that out. So this matches what we would see. And when we give them this drug at either I did both a low dose and a high dose, it reverses the effect, right? Suggesting that allopregnanolone at the neurosteroid binding site on GABA-A receptors is necessary for this phenotype, and we can block it otherwise. Okay, so back to our working model. So we have our PVN with the peri-PVN projections, which inevitably we're getting anytime we do kind of RNA-seq or attack-seq. Anytime we punch the PVN, we're getting those. And now we think it's an increase in GABA. So what that increase is still to be determined, right? There are a lot of ways that we could get increased GABA tone. And that allopregnanolone is part of driving that increased GABA and interacting with this background of an altered chromatin state that's setting up the PVN of these females to respond differently, and males, when allopregnanolone is available. Okay. So kind of for the work I've shown you so far, before I talk about what kind of future directions, so we have a translationally relevant model here. So when humans and mice with pubertal adversity show long-term disruption of their stress response. It's evident during pregnancy and postpartum, and in the mice it's only evident during pregnancy or postpartum, or if you give them hormones. It's sex specific. So this is not a sex difference necessarily, but it's sex specific in that males never experience allopregnanolone in their lifetime, so they're not at risk. They have that first risk factor of the puberty stress, but they don't show the phenotype because they don't have the second risk factor of going through the hormonal changes of pregnancy. But when they encounter them, they show the same phenotype. So one of the things I didn't, I always debate about whether to include this, but I'll just kind of preview it here. This is actually experience dependent. So when we go back to risk and resilience, I'm also deeply interested in what makes individuals resilient and what kind of experiences can do that. So another kind of third group that we always have, but it's kind of difficult to wedge into a talk, is that if we take the animals during puberty stress and we let them have social interaction between the stressors, so they get to go back with mom and litter mates in between the stressors, but they experience the stressors alone, it prevents the effects that we see. And it's amazing. Now, anyone who's studied risk and resilience knows that resilience is not the same thing as being in control, right? So on all of these measures, sometimes they look exactly like controls, and sometimes they're their own third group. So it becomes, now you're figuring out two mechanisms, basically. So it can be difficult to bring up in a talk, but I'd be happy to talk to anybody about that in questions or later. And we also think this is due to GABA reprogramming. So that what puberty stress is doing is it's changing the GABA system, and that pregnancy unmasks this by increased allopregnanolone. Okay. And so this is set up, for my grant and my future directions, this kind of overarching hypothesis that I've kind of outlined already. But that stress dysregulation during pregnancy and postpartum is an interaction of changes puberty stress makes to the PVN with increases in allopregnanolone. And importantly, that this is not good for the animals, right? So in the humans, we contextualize this with the Edinburgh Postnatal Depression Scale. And in the animals, I've done things like pup retrieval. Pup retrieval is super hardwired. It's very difficult to disrupt their pup retrieval with a mild stress early in their life. But what we know about stress, vulnerability to stress, is that it's usually two hits, right? There's a two-hit hypothesis. You need a genetic risk factor and stress, or you need stress early in life and stress later in life. And so the idea here is that these individuals are at risk for further dysfunction, perhaps if we push them during pregnancy when they're vulnerable. So the first project has been to identify this epigenetic reprogramming. And so that's been the ATTAC-seq and further analyzing that and trying to find ways that we can manipulate the epigenome and perhaps prevent this from happening in puberty. So the second project is to demonstrate how is allopregnanolone doing what it's doing. Is it really at the GABA A receptor? That's the hypothesis, but we have kind of some preliminary pharmacological data there. How is that happening, and is it allopregnanolone that's driving the changes we see in the chromatin? So we see those big differences. Is it really allopregnanolone that's driving them? And how is it doing that? And then finally, again, is to demonstrate that the latent, so we call this kind of latent stress reprogramming that becomes unmasked by pregnancy puts them at risk, right? And so when we think back clinically, so it's one of the benefits of working with clinicians is they're always like, that's really great, but how am I gonna help my patients? Sometimes it's just knowing that you have risk factors, right? So if you can assay women and bring them in during pregnancy and ask them about their life experience, that's one way we can just put people on notice. Like, you're at increased risk, we need to keep an eye on you, et cetera. One day maybe they'll be, so there is also, so on the allopregnanolone front, there's been a pharmacologic, so brexanolone to treat postpartum depression, and that's an allopregnanolone-like substance. And so there's something here, I think, to allopregnanolone being really important for these phenotypes. Okay, so just a few details about kind of what we've been doing and what I'm continuing to do on this first project. So we've done the ATTAC sequencing. And one of the benefits of ATTAC-seq is when people look at changes to the chromatin, they often pick their favorite histone mark. And that's fine. But so what we've seen we've done some histone mass spec. And so this is an example. So this is from a separate study where I did looking at the puberty brain. So kind of all my work is focused on pregnancy, but I'm really interested in what's happening during puberty, so there's a lot to go on here. But this was actually the prefrontal cortex, so kind of slightly different project, where we looked at just sex differences across puberty. So postnatal day 28 and 35. And when we do things like histone mass spectrometry, where you can actually unbiased look at all of the histone marks, instead of just picking one that has a good antibody or two that have a good antibody, what you find is that it's not one mark. It's many marks. It's often many marks in concert with each other. And so it becomes a little more difficult to say, we're going to pick X mark. You're kind of missing everything else over here. And so I kind of like to get a profile of that in our animals. So we haven't done that yet. But this is helpful to kind of show you where the big changes are. Okay. So our immediate early genes have been the nice readout, and so it would be nice to link our chromatin modifications to altered immediate early gene expression, but that counts on you being able to pull something out of the histone mass spec. So if you do find a mark, so some kind of histone mark, so H3K27 trimethylation, it's a popular one, in our lab anyways. What you can do is you can pull down that mark and anything that's associated with it. So this is chromatin immunoprecipitation. And so you get an antibody for your histone mark, you pull that down, and anything that's attached to to, and then you can look at what is that mark near in your chromatin. Is it regulating the genes you think it is? And because we already have genes that we're interested in, we don't have to do ChIP-seq, right? So we can do ChIP-PCR. So pull it down and look and see if our mark of interest might be related to our immediate early gene changes. There are also some things that seem, like say epigenetic reprogramming, this is kind of fairly basic, but understanding what the tone is, right? Is there actually more GABA? So VGAT suggests there might be more GABA because its role is to bring GABA into the synapse. But we haven't actually measured that yet, so that's something we can measure. We've done it previously, so this is again from the PFC of males and females during puberty. So that's something we can do with our PVN as well. So I'm also been working with one of our technicians who's really great with, she's been helping me with imaging, which is great, to do immunohistochemistry for VGAT, so protein. So again, when we look at these things on the gene level, there are lots of levels between it being a gene and it being here out in the protein. And so it's important to assay these things on multiple levels. And it's the same with the omics, right? So RNA-seq or ATAC-seq pairs really well with things like metabolomics or proteomics to kind of put the pieces together of, is it linear or are things getting changed in that process? And so immunohistochemistry can help for that. So this is a preliminary VGAT stain here. And importantly, what we really want to know is how do these CRF neurons respond to input? Because that's what we're seeing when we stress them, right? We're giving them this broad stress, but we're really interested in what has changed about how they respond to GABA input. So this part is for sure, this part is kind of me hopeful. But we're collaborating with Scott Thompson, who's an electrophysiologist at the University of Maryland, to do electrophysiology. So we're going to do, not I, our collaborators will slice, and we have a CRF reporter mouse, so we can look at the CRF neurons and do things like put allopregnanolone on, take it off, and kind of do some of those manipulations that are really difficult to do in vivo to kind of point us to future directions about what's going on. And then what would be really cool is to do patch-seq. So you record from your neuron, and then you suck up the contents, and then you can sequence it. And you can really see, do the things that happen physiologically match with what you think is going on at the gene level? And I think that's a really important, and it's difficult, but I think it's a really important jump to make to see if it really matches what we're seeing. So we're also continuing clinical collaborations. So one of the downsides of Tracy moving and then Neil also moving is that you kind of lose your cohorts and your collaborators. Now you have two people starting new labs. And so right now we're working with Tanya Jovanovic, who's, well, she's also actually moving to Wayne State, but she's been the head of the Grady Trauma Project, which is in Atlanta. And the Grady Trauma Project is amazing. So every person that comes to the ER basically gets screened and put into a study. So they have a wealth of information. And so when we talked to her, she said, we have samples in the freezer. And one of the really interesting kind of questions going forward, both for the animal model and in humans, is puberty actually a sensitive period? Is there something special about experiencing stress during this window? And so they have cohorts where adult women that have come through, so they're not pregnant, but this is a start. They've had trauma in one of 3 developmental stages. So prior to age 13, from 13 to 17, or 18 and older. So we can start to look in humans, is there any signal, anything different about experiencing trauma in any of these different age brackets. We also try to focus on things we can measure in humans and mice. And so I don't know if anyone's seen, so there have been some circulating cell-free mitochondrial DNA papers recently showing that in plasma, I don't think anybody knows what it's doing, but it's a good marker that it's been associated with increased suicidality, more circulating cell-free mitochondrial DNA, increase in depressed patients, and actually responsive to treatment. So it's high in depressed patients, and when they're treatment responsive, it comes down in their plasma. So these are really good biomarkers that we can do in humans and mice to try to understand what's going on. And then also we have an interest in extracellular vesicles as another communication mechanism. So just like briefly, EVs are ways that the body communicates in very long distances. So these are packages of proteins, and they have often mRNA in them, and they can go from tissues very far away from each other to signal what's going on. And so we've been examining what's in extracellular vesicles. Also very relevant for pregnancy because the placenta makes an amazing amount of extracellular vesicles. And so how that's signaling to the body to accommodate pregnancy, and also how that might go wrong if someone said early life stress. So to like kind of back it way out, what's the lab going to look like? I'm going to continue to investigate cellular and molecular mechanisms of experience-dependent resilience and vulnerability, and how to stress, and continuing to understand how sex, developmental stage, and the type of experience shapes an individual's trajectory. So these are complicated models, but we're getting closer to kind of what we can ask in humans with multiple hits and across the lifespan. So hopefully continue to collaborate with basic and clinical researchers. So a lot of this work has been with collaborations with others, and I've really enjoyed that. And then to continue teaching and mentoring. So again, a lot of the best experiences have been teaching students how to do something and then having them get, I mean, it's always better when everyone has a successful result, but when they complete an experiment, that's always good too. Okay. So with that, there are many people to thank. Tracy, who's been a fantastic mentor and very supportive of anything I've wanted to do, and including many people in the lab, some of whom who aren't there anymore, but so Allie Rogers is a super talented grad student, kind of helped me start this model. Patrick Kane and Victoria Meadows are new technicians who have kind of like jumped in the project with real excitement, and they're helping me out, kind of hopefully wrap things up, as well as many other people in the lab with great expertise and help. Also Neil's group, who was really fantastic in starting this collaboration. Our kind of brand new collaboration with Scott Thompson's lab, and Tony Cole's an MD, PhD student who's gonna be doing that patch clamp. And then at Penn, we had the Center for Host Microbial Interactions that had this is where we learned how to do sequencing. Dan Biting runs that. And of course, funding. And for all of you for being here today and for inviting me to come. Thank you very much. Yes.
In the animals where you stressed them, how soon after they were stressed during postpartum did you collect their tissue and run your RNA sequencing?
So the stress is always during puberty, and then now that's from 3 to 5 weeks. And now they're 10 weeks old when we breed them. So the RNA sequencing was done on E18.5, so the day before they're about to give to birth. Okay. And we've done the HPA early and late, but we were focusing on late because that's where we see the biggest differences for the mechanistic kind of work.
I guess my follow-up question to that is, I mean, you had kind of hinted at this during CFOS expression and looking in that area in these animals, because it suggests, your data suggests that you have a certain population of neurons that are hyperexcitable.
Yeah.
Right? And that would help you identify which population of neurons those are. And is your electrophysiology that you had discussed actually going to evaluate that in those neurons?
So we haven't started the EFIS yet. So this was one of the things that, you know, when a lab moves, things kind of get derailed. So we've just now, and we also needed the reporter mice because we wanted to focus on CRF. Although we also have GABA reporter mice as well, if it ends up being, because it could be the GABA neurons that are changed. So we haven't been able to address that yet. I do have some, so the CFOS has been kind of puzzling because I did what I thought was gonna be like a slam dunk experiment, where I was like, I'll just do FOS. Like, this is different. It should be different at the protein level. This is gonna be great. They're not gonna be different before they're pregnant, and in pregnancy they're gonna be different. And it's the exact opposite on the protein level. So I was like, I don't know what to do with this data. So the way that I've been thinking about this, and there are more experiments to do to see if I'm thinking about it correctly, is that because they're hyperexcitable, I think on the protein level, they're perhaps ahead of the curve, right? So by the time we get out to our normal, this is 90 minutes after our restraint stress, where we'd expect to see kind of we're peaking, they're already coming down because they're transcriptionally already available. That's my hypothesis for why this looks like it does. I don't know if it's a very hopeful hypothesis. But I do think one of the things that helps, what we can kind of salvage from this, is that they're already different before pregnancy, but there's some disconnect, right? So like they have a difference in FOS, but they don't have a difference in glucocorticoids, right? So why is that? So it also kind of identifies a circuit, and if anyone knows anything about CRF, these are kind of all CRF regions. So CRF might actually be important in what GABA's, the hypothesis, how GABA is regulating CRF. Yes?
About this, are we looking at the numbers of cells relative to the?
It's the number of cells relative to the density.
Okay.
Yeah.
So have you looked at the relative intensity of the cells?
Yeah, so the first thing I did, right, was, so the intensity of the FOSS cells or the host, the relative intensity. So this is all number of cells. No, I haven't.
It's not.
I haven't looked at the number of intensity. Right.
Since we're talking about immune staining,
Yeah.
gets that to my second question. Have you done CFOS plus CRF staining?
Have I done CFOS plus CRF?
If it's the CFOS cells that are actually.
Not yet. Yeah, not yet. So that's a really good point. So in with the VGAT is also to do things like CRF to try to understand, is it the CRF neurons? So it's
very heterogeneous.
Very heterogeneous. Right. Yes.
RH.
There's so much there. It continues to amaze me. Yeah, for this tiny brain region, there's like 3 of everything. And there are a lot of different kinds of things there. Yeah, so, I mean, CRF is the most obvious target, which is why we've started there. I can't say that I have any good evidence other than the hypothesis that I have for it. So with the VGAT staining, we'll also be co-staining for CFOS and other markers as well to see if those are really the neurons that are changing.
What percent of the cells in PVN are CRF positive?
Oh.
And does that change?
Does it?
Pregnancy to pregnancy?
I don't know if anyone's looked at that specifically, but I don't think that the number of cells change. I think it's the way they respond. Yeah, and the kind of the profile. I don't know the percent. You're making me feel like I'm at my prelims or something. Oh man, I'm gonna be in trouble. Don't tell Tracy I didn't know the percent. No.
I work at thyroid hormone, so
okay.
honestly, my interest in PVN is more on TRH.
Right, right, and it changes. So like, I know anterior to posterior, it's different, right? So they're more. You don't know either. All right,
I don't know the percent of cells.
All right, well, I don't feel so bad anymore. So I know it changes anterior to posterior as well. I mean, it's a very heterogeneous structure. And so when I started doing this, people, it's like, why don't people study the PVN? It's so interesting. I'm like, I know why people don't study the PVN. It's complicated and it's tiny. So it can be really difficult to do assays on because there's just not enough stuff there. So we've been working on doing some Westerns as well. What I was hoping to do was Westerns for synaptosomes, like really pull out for GABA. So if it's GABA A, like are there, because one of the things that could be happening is there are changes to the GABA A receptor conformation that are changing how sensitive it is to things like allopregnanolone. Like, oh, it would be really cool to get synaptosomes and then look at Westerns for different GABA A receptor subunits. That's impossible with one PVN. I mean, you need like to pool 8 animals, and at some point it becomes, it's not a good use of the animals necessarily. So I'm looking at other methods for doing that, but it's definitely a question. Yeah, thanks. Yes.
So when you did stand for VGAT and you like looked at the number of GABA neurons?
So did we stain for VGAT? So it's in process, right? So we're working on that. So we haven't actually quantified it yet. One of the things about VGAT is it's everywhere. Yeah. So I don't know if you saw in the picture, all that green, that's all VGAT. It's really ubiquitous. So what we're gonna do is kind of look for puncta. So you have to get down, and this is why the confocal is necessary to kind of look at CRF neurons, and are there more VGAT if the CRF neurons are in the, CFOS are in the CRF neurons, right? Is it changing the number of puncta? There actually seem to be more VGAT at these synapses that we're interested in, but I haven't completed that study yet. It's a good question.
Because like you're looking at like your hypothesis on there being like an allosteric modulator that's like affecting GABA, you don't expect it to be like a permanent increase or decrease in the number of GABA neurons like following pregnancy.
Well, it could be a change in so it could be we don't know, right? So one of the things is that I don't hypothesize that it's a difference in the amount of allopregnanolone. So we've done allopregnanolone peripherally, which isn't, it's as well as we can do, and it's not different. So we've seen that in all the hormones we've profiled, that stress doesn't change the amount of hormone, but it changes the sensitivity of the individual to that increase in hormone. So it could be GABA receptors, it could be GABA neurons. There are a lot of players in the system there, and we haven't quite pinpointed which one it is yet, but there are a couple possibilities for that, yeah. Yeah.
So in your ataxia,
Uh-huh.
you're showing in your versions that the stress like basically opens them up.
Yes.
Because in pregnancy, there's like a couple different things going on. So do you still see that like overall increase in opening, or do you see like a mixed bag of
Oh, this is the question I didn't want to get on my poster at SFN either. Now, it's a great question. It's really more clear-cut in the virgin females where it's more open. The changes are really, really dramatic in pregnancy, and some of them are that things are closing. And it's also, things can come out differently how you do the analysis. So this was the effective pregnancy on each individual. So another good way to do the analysis is to compare control and stress females during pregnancy to get a more direct answer to that question. But I mean, if you saw the plot that I showed, right, in CVS females, so this is increase and this is decrease. And what pregnancy does to CVS females is decrease a lot of stuff. So it's like anything. It's not a perfect fit. And partially that's because probably immediate early genes do a lot of stuff, and this is just an indicator. So I was actually really excited that this turned out at all, because it felt like kind of a big swing based on immediate early genes. But yeah, it's a little more complicated during pregnancy, which is another reason people don't study pregnancy. It's all complicated, but really interesting. It's a good question. Well, thank you very much.